phosphate-free detergent or hydrochloric acid baths that you can use, first of all, before you rinse it with Millicue water. And that rinsing has to take place 3 or 4 times in order to make sure that there aren't droplets of contaminated water that still remain within this container. When you go out into the field as well, you want to make sure that this container is rinsed a couple of times with the water that you actually sample, because clearly if you have water, and it may even be distilled water that's sitting in the bottom of a tube, and you then fill up that tube, then of course you've diluted the sample. Or vice versa, if that water is contaminated, you've then is at a higher concentration, you've then contaminated the sample. So the essence of this is that if you rinse it a couple of times with the water you're going to sample, then any of that contamination is hopefully negated. So there are a variety of containers that we'll go through today that may collect the sample, and they include the Schindler-Pertalis, they include depth integrated tube samplers, which is basically a hose that will integrate some depth of the water column that you wish to sample. The Schindler-Pertalis will take a discrete depth sample, and the Van Doorn will take another discrete depth sample for you. So they also should be rinsed quite thoroughly, at least with the water that you're going to sample, before you actually do the sample, before you collect the sample. And so then you'll put it in a tube, and generally we filter on site. Okay, so one of those samples will be kept as it is. The water goes straight into the tube, and it will be analysed for total phosphorus and total nitrogen. With the other tube, that will go through a filter, which is typically called an 0.45 GF, commonly GFC filter, sometimes GFF. There is a subtle difference between these glass fibre filters. This is the actual particles that go through these filters are up to 1.2 micrometers in size. So in other words, while it says 0.45, it's actually a 1.2 micrometer particle that can be up to 1.2 that will go through the filter. And so we filter first of all, and again, that apparatus that we filter with should have been rinsed thoroughly with the water we're going to sample with before we do the sample, and then we collect the dissolved, so-called dissolved material. It's not truly dissolved. It does have these fine colloidal type particles that are less than 1.2 micrometers. And for that sample, we will be analysing such things as phosphate, nitrate, nitrite, and ammonium. And I don't want to give you a long lecture on nutrients, but when we express these, commonly they're expressed as PO4-P, or alternately as milligrams P per litre, where the concentrations we express are actually as the P atom and not the whole PO4 molecule. So it's a subtle point of difference, but it's a very important point of difference that you actually express these concentrations in terms of the P atom and not the P molecule generally. The other little gripe that I have is a lot of people call it ammonia, and generally what we're analysing for are the ionic forms of these nutrients. It's actually ammonium, NH4+, that is analysed for. So, the objective of once having collected these samples is to put them on ice, 4 degrees Celsius, slow down any biological processes. The filtered sample should be reasonably stable, but especially if it's on ice, 4 degrees Celsius. Get it back to the lab and commonly freeze it. It's generally not possible to have the nutrient analysis done immediately because of the setup time that it requires for that machine. So it goes into the freezer, and maybe someone wants to cover, in Louise's absence, just the log-in procedure. So we have a log-in procedure. We give a barcode to the sample, and that should enable us to retrieve it at any time into the future when it's in the freezer. And following that, we defrost the samples, and we have our totals. That's TM and TP, and we have our filtered, which is going to be our nitrate, nitrite, ammonium phosphate. These ones we have to do a digestion. And again, in doing that digestion, we run the risk of contamination. Okay, so we need to make sure that whatever we use in preparing that digestion solution and any equipment that we use, we want to make sure that it's absolutely clean, because you have to keep in mind that in most cases we're dealing with concentrations of micrograms per litre, which is parts per billion. So it's very, very easy to cough on the sample, to sneeze on it, and to immediately contaminate the sample. That's the sort of thing that we're talking about in terms of our ability to contaminate the sample. So that digestion is designed to take all of the forms of nitrogen and oxidize them through to nitrate, and all of the forms of phosphorus and oxidize them through to phosphate. So in other words, even material that is in particulate form is oxidized through to nitrate and through to phosphate. That then enables us to put all of these samples into the analysis plan. But the key part of doing that analysis is to be able to prepare standards. These standards are designed to have concentrations that more or less replicate the range of concentrations that you see in your samples. What do I mean by standards? Those standards will be prepared so that we have concentration here. And we have essentially colour here. Because what we do in the analysis procedure is we add reagents that give the sample a certain colour, and that colour is basically a distinct colour which tells us about concentrations that we may have. So for example, the colour for phosphate, given the reagents that we add with molybdenum and so on, is blue, the ammonium is green, and the nitrite is generally red. And so we've got a machine that is basically able to pick up those colours. So we prepare a set of reagents of given concentrations. We'll have a range of concentrations here, typically about 5 or 6 standards of different concentration, including, most importantly, blanks, which are designed to essentially set our zero case. And so in the case of this digestion, where we've got samples, we treat the standards in exactly the same way, and one of those standards will actually be a molecule water standard. And so ideally what we get is a certain colour the machine tells us, and hopefully a nice straight line that corresponds to our known concentrations for each of those standards. We then put a line through here, and generally we get an R squared for that line, the Pearson correlation coefficient of 0.99 or so. If you're getting less than that, then maybe some cause for concern. There's always a little bit of variation here. And so then we run our samples, knowing that we've been able to get our standard line. Our samples colour up to a certain amount, and we then can use that line to give concentrations of our samples. So that procedure is probably the most critical of all of the nutrient analysis, because if you make a mistake with the reagents, then in most cases it's just a case that the colour won't be as intense. If you make a mistake any mistake at all with the standards, if you dilute by 10 or you get your concentrations too strong by 10, and so you've ruined everything. So the preparation of the standards is the absolutely key part of the whole analysis. Okay, very simple lesson there, but it's happened many times. I'm sure you'll see some samples that have been listed in here. Don't use a paper label, it will get wet and come off in a freezer. Don't use pencil or pen, use permanent marker only. If you need several tests, use 50 ml Falcon tubes. Okay, so that means that you can take do multiple analyses with one tube. On the other hand, you may want to duplicate it at this stage. And I can't make a firm recommendation on it, but when you're doing your pilot type studies, it may be useful to have that capability to just go and see what the variations between samples. 15 ml tubes are fine, but Louise suggests take a replicate set. Alternately, you can use these for your totals and your filtered, and they actually give you plenty of volume to be able to take your samples, provided, of course, that everything works smoothly. So again, Louise says use permanent markers only. In this case, there's filtered samples in blue, totals in black. That's just to make it easier, I guess, for retrieval. Ideally, prepare labels before you go into the field. It's a heck of a lot easier to have everything labelled up before you head into the field. And vital information on the tube, lake identification, depth or site, date, and identify clearly if total or filter. Additional information, you could put it on all of this within a bag so that you can retrieve it easily on it for a given day. I think that covers most of it. The analysis part, we have two means of analysis. One of them is called the flow injection analyzer. It's been with us for quite a long time. And the other one, coming up here, what's it called? The new one?
With the screen.
The screen analyzer. Heck with keyboard.
And I think that we are getting some extremely accurate results out of the discrete analyzer, but these two, when operated correctly, and when you prepare your standards correctly, will give you actually fantastic results. With the sort of only caveat that when you do this digestion, you do get contamination. Don't ask me where it comes from. I think it probably comes from maybe the tubes themselves. It might come out of the atmosphere, and it may be the chemicals also that we use in the digestion, which means that this baseline here, this concentration of 0 down here, does have a certain colour. Okay, so often our standard line actually is like so, with a certain colour given to that blank. So that means, or the 0 one, so that means that it's very important when you do these standards to have at least probably 2, 3, 4 to be able to give you that 0 in that background case. So the analysis, as I've mentioned, is about preparing some reagents plus sample which goes to colour. As I mentioned, you want to prepare those reagents accurately, but it's not as critical as preparing the standards. So the reagents, all that you get is a change in the intensity of colour if perhaps you've made a very, very minor mistake in terms of the mass of the material, for go into this part in detail because Bruce is much better than me to be able to cover aspects of that. But what I will say is that both of these machines require or analyse for nitrite. So in other words, if you don't alter the sample goes directly in, then you get nitrite. If you put the sample through a cadmium column, then you have nitrite, and you will have reduced the nitrate down to nitrite. So you analyse for NO3 plus NO2, and so as a result, your NO3 is equal to your NO2 plus NO3 subtract your NO2. Okay, so any questions on this?
With the concentration and if you're taking multiple samples, say from the stream, you get a rain event, which is theoretically increasing the nutrient concentration. Can that sort of be off the scale, and so you may have to be working on two different scales?
Yeah, Bruce might be good at that. Answer that than me. It's about switching from the, I guess, from the low to the medium and the high on the discrete, isn't it?
Yeah. Yeah, with the discrete, it has a certain, and the FIA has a certain range of optimal concentrations it can detect for. If it's too high, there's going to be some dilution done to bring it back into that range of detectability. With the discrete analyzer, there's some automation to that within a certain degree. But with the FIA, once you've reached that ceiling, you've got to manually dilute it. Otherwise, you just go beyond the detection abilities of the machine and get rubbish results. For the FIA, it's roughly probably upper limit about 4 milligrams per litre of any analyte.
Which would mean you then need to start doing some dilutions on it and potentially dilute your standards, which just runs a greater risk of the analysis not being quite as good. And I'm glad you mentioned that too, Grant, because just to reiterate, your concentrations of your standards should be at least the upper bound of your samples. So if you have any idea, that's why the pilot sort of work is quite important. But you're much better to prepare standards going much higher, and if your samples fall into a lower range, just throw away the top standards. And the other point is we usually prepare mixed standards. And those mixed standards, you have to be very careful again about mixing phosphate with nitrate and with ammonium. So typically our mixed standard might have as a sort of stock solution 10 milligrams per litre of phosphate. Sorry, make that one. Let's say 10 milligrams per litre of nitrate and 10 milligrams per litre of ammonium. Now what that means is that we've got a ratio of nitrogen to phosphorus in terms of mass or concentration of about 20 to 1. And that's what most people will see with a large number of their samples. That mass ratio will be about typically 10 to 20 to 1 in most samples. So just keep that in mind, because it's no good throwing in a 10 milligram per litre phosphate in a mixed standard with say 10 milligrams per litre of these. Normally that ratio will be 10 nitrogen to about 1 phosphorus, or 20 nitrogen to 1 phosphorus. And that's, for people that know the Redfield ratio, that's a middle ground roughly for the Redfield ratio. If you don't prepare these correctly, it limits your ability to generate 3 of those curves for each one of those